Hey everyone, and welcome back to Tutorism, your go-to spot for GCSE Biology revision and exam practice. Today we're zooming in literally on the principles of microscopes and how to prepare a specimen slide. So let's get microscopic. By the end of this video, you should be able to describe the structure of a microscope and describe how to prepare a microscopic slide and use the microscope to observe a specimen. Magnification is the process of enlarging the appearance of an image, making something look bigger than it actually is. First up, let's break down the parts of a light microscope. You'll need to know these for the exam. Firstly, we've got the eyepiece lens, which is usually ×10 magnification, and this is where the observer looks through to see the magnified image. There are 3 objective lenses, typically ×4, ×10 and ×40, and you can change these lenses to adjust the magnifying power of the microscope. Next we have the stage. This is where specimen slides actually sit, and the stage clips help to hold the slide in place so it doesn't slip off and break. Next we then have a light source. This helps to illuminate the specimen so we can actually see it. Sometimes instead of a light, microscopes have a mirror there so you can reflect light into the eye. Finally, we have the coarse and fine focus knobs. These help us to improve the resolution of the image, to sharpen it so we can actually see the specimen more clearly. Now let's talk about how to prepare a slide step by step. Firstly, place a thin layer of your sample, like onion cells for example, on a clean glass slide. Then add a drop of stain, for example iodine if you're using plants, or methylene blue if you're using animal cells. Remember for the exam that stains actually help to highlight cell structures, because cells are mostly water, so without them it's like trying to find a salt crystal in a tub of sugar. You simply won't be able to see the cell very clearly. For step 3, we're then going to gently lower the cover slip over the sample using a pair of forceps. This is going to help stop the sample from drying out. Make sure that you do this carefully so you avoid trapping any air bubbles, because these can actually mess with your view. Now it's time to observe your specimen, so we'll follow these steps. Step 1 is to secure your slide onto the stage with the stage grips. Step 2 is to ensure that you actually turn on the light source, otherwise you won't be able to see a specimen. Step 3 is you're then going to start off with the lowest objective lens, which is usually ×4, and this is going to give you a total magnification of ×40. A bonus tip is that we always start off with a low magnification and work our way up. It's like trying to find somewhere on Google Maps. You don't ever start at street view. Okay, we start as an overview and then we zoom in. The next step is to then use the coarse focus knob to try and bring the image into view. And then finally we're going to switch to the fine focus knob so that we can increase the resolution and give us a nice sharp image. Right, so a tip to help you remember is the phrase "great scientists study cells." This can help you to recall the steps to prepare a microscopic slide. Great stands for glass slide, scientists stands for stain, and cells stands for cover slips. So this is what you need to be able to prepare a specimen slide. Next, we've got the acronym SLOC. This can help you to remember the steps involved in setting up a microscope. The first step is place your specimen on the stage. The second is to turn on the light. The third is to adjust your objective lenses. Remember, we usually start at ×4 with the lowest magnification. And the final step is to adjust your knobs, that being the fine and the coarse focus knobs. This is an easier way to help you record the steps. So now it's time for us to do some guided practice together. So we're going to have a look at an example answer to an exam question and discuss why it was awarded marks or why it didn't get any marks. So the question is, a student was investigating mitosis in the roots of an onion plant. Describe how the student could prepare a microscope slide to show mitosis in the growing roots of an onion plant. This is a 4-mark question. The student wrote, cut a piece off the root of an onion and peel off a thin section of cells with forceps. Place down onto a glass slide, add methyl blue stain, then observe with a microscope. Now, this student only achieved 1 out of 4 marks, so let's actually look at why that is. The first bit says, cut a piece off the root of an onion. Now, this would not be awarded a mark because the question specifically says you are investigating mitosis. That means we are looking for the part of the root where we're going to have lots of cell division taking place. Now, which part of the root has lots of cell division? That's actually the root tips, because that's where the meristem tissue is. So therefore, we have to replace that if we want to get that mark. Next, the student said, peel off a thin section of cells. So this is correct. They would achieve a mark for that. Now, we're not actually going to give them mark for saying place down onto a glass slide. And the reason why that is because it's in the stem of the question. The question says how to prepare a microscope slide. So you can't always get marks for repeating what's in the question. So instead, we have to look at the next point, which is about the use of a stain. Now, it's great that the student said we're going to use a stain, but unfortunately, they have said the incorrect stain when we're looking at plants. When we're using animal cells, we can use methyl or methylene blue stain, but because this is a plant cell, we need to use iodine. So we need to change that to get that 3rd mark. Lastly, the student didn't discuss the final step of slide preparation, and that is adding a cover slip. Okay, alternatively, they can say add a drop of water, and the reasoning for this is to stop the cells from drying out. This answer would now be 4 marks. Right, now it's time for you to have a go at applying your knowledge. So I'd like you to attempt this exam question. The question reads, a student cut a piece of onion and placed it on a microscope slide. The student then placed this glass slide on the stage of a light microscope and looked through the eyepiece. The student could not observe any cells. Suggest 2 changes the student could make to improve the view of the onion cells and give reasons for your suggestions. So this is a 4-mark question, which means that we're going to be awarded 1 mark for each suggestion to a change that the student should make, and then 1 mark for a reason for those suggestions each. Okay, so if you make 1 suggestion and then you give a reason and you do that twice, you should achieve 4 marks. Pause the video and have a go, and then once you're ready, press play, and then we'll go through the mark scheme together. Right, let's go through the mark scheme. So the first mark is for saying turn on the light, and the linked 2nd mark to that is to see the structures clearly. So if you say this sentence, you can be awarded 2 marks. Similarly, you can also be awarded 1 mark for saying adjust the fine or coarse focus knob, and another mark for saying to make the image clearer or to improve the resolution. You can also get another mark for saying add a thin layer of cells, and the linked mark for this is so light can pass through. So that sentence would be another 2 marks. The next one is use a stain for 1 mark, and the linked mark is to make cell structures visible. And then next we can also say use a bigger magnifying lens, and the linked mark for that is to make the image clearer. Okay, so essentially if you say any one of these sentences, that would be 2 marks. So if you say any 2 of these sentences, you will get 4 marks on this question. Right, so you should now be able to describe the structure of a microscope and describe how to prepare a microscopic slide and use the microscope to observe a specimen. Thank you very much for staying and watching this video. I hope that you've learned something today, and do keep a lookout for my next post as part of the Microscopes and Magnification series. Check out that growing playlist on my channel. Enjoy the rest of your day.